All right, hey everyone, welcome back. Today we're gonna keep looking at this new add-on for Blender called BioBlender. Today we're gonna look at DNA and RNA and how to implement these two into our general scene. All right. So what we're gonna do here is first, I'm gonna import one of our main proteins that we're gonna work today, 1TAU. That's gonna be DNA polymerase. So since we're gonna be working with DNA, I would be good to have a DNA polymerase here. I think that's pretty cool about the add-on and the way things work is that everything is to scale. So everything is following atomic scale. So every object we import here, DNA, membranes, proteins, molecules, everything's to scale. So everything should be resembling what we would see in real life. In this case, I imported DNA polymerase here. Let me just put it in the selection to cursor so I have it in the world origin. This is just for convenience once I get the DNA. Okay, so let me just rotate it. So if I rotate it this way, and let me also rotate it this way so it's aligned here. Alright, and let me just move it up. Oh, let me just move it up a little bit. Okay, so let's give it a material. To do that, we just select it, and we can do that real quick by applying here. So now that we have that material, we can maybe also change the color slightly. Let's go to we can do an orange. Okay, let's do orange for today. Alright, so now that we have DNA polymerase, and I'm gonna move it in fact a little bit over here. Alright, so now that we have DNA polymerase, we're gonna talk about today about DNA/RNA. So here we have the type that is basically gonna we're gonna have RNA or DNA. That's our two options. Then visualization type, we only have two options, surface or cartoon. And then we have different predefined paths for our DNA to follow. Alright, so the surface is the usual representation that we are used to seeing in proteins, so very similar. So if I go ahead and for now let me import them as without any curve, and if I bring RNA, this is what it's gonna look. So let me bring RNA as a surface over here. If I bring DNA, this is what it's gonna look like. So we do see that double helix shape. Let me just do this one more time for a cartoon version. So if I go to cartoon DNA and bring that, this is what it looks. This is a cartoon version that I modeled, so it's not based on PyMol. In this case, it's just it's for it to look better, and it already has information regarding the colors. So it has that color data regarding the basis. And then just this last one, let me just bring RNA. So there we have RNA, DNA as a cartoon surface, and already brings colors. Alright, so let me delete these objects, and now we're gonna go into this final setting here, which basically gives us a predefined shape for our DNA. So let me hide this protein for now. Let me bring a vesicle. Let me turn this cross section to 0. Once I have the cross section to 0, let's say we wanted to represent that usual image that we see, that it's kind of a spiral DNA around inside a virus or something that we would see like. So we can go ahead and select, let's say RNA in this case. Let's go to spiral, and then we can select, let's do it as a cartoon in this case. So we import that, and as we see, if we go to the shader, to the cycles, then we do see DNA being coiled following that spiral pattern. Another way to visualize this is if we do surface in this case. So let me just go here. Let's do surface of this, and let's maybe bring DNA that has a little bit more volume, and this is what it would look like. We can select a material again, apply the real material, and then we can go ahead and it would be orange as the one we defined for our polymerase, but we can always change the color hitting the number 2, and then making it maybe purple if we want some pink DNA. So that way, if we look at this from far, this is what it would look like. So that is an easy way to get DNA inside our scene. Let me delete this again. We could also have these other, so we could have this nucleoid type of shape. This would be more like what we would find inside of bacteria. So we get this coiled ball of DNA just by doing that. And then there's other different shapes that we can get. We can also have a twist. This is basically just gives us a coiled up DNA. Another one, last one that we might want to look at, it's nucleolus. So we have one or two. This is just going to give us again a different shape of DNA that it's yeah. So now let's go to the last mode, or one of another mode of this importing this DNA. That's going to be free. And what we're gonna do here is bring back our or show back our polymerase here. And now what I'm gonna do is have DNA be imported as a cartoon. So I'm just gonna change this to cartoon, and I'm gonna select free. And as we see here, we get our DNA here in our scene. What we're gonna do is, instead of grabbing the DNA, we're gonna grab this curve that's called free. We're gonna go into edit mode, and now what we can do is press E to extrude. And we're gonna be moving it around. So let me just go ahead and move this around here, move this around here, and as we see the shape of our DNA polymerase, here at the bottom is where DNA would come in. So somewhere, let me rotate this a little bit. This is not gonna be perfect because it's being done quite fast, but as you can see, it, even if I'm doing it real fast, it works pretty well. So let me just rotate around this way, move them this way, then I would have to move some of this one down so there is no intersection. But as you can see, DNA usually this is the way DNA would go inside this DNA polymerase. And as we see here, I just have to move here inside. As we see here, as I was mentioning, the scale works exactly as it should be. It's just, you just have to play with organizing these so it's following the exact shape of it. But yeah, that just takes a little bit of time rotating and moving around these vertices. But as you can see, you can get all sorts of shapes, and I moved that too much over there. But as you can see, you can get all sorts of different shapes just doing it this way, and then you already have a nice visualization of DNA going through your DNA polymerase. We can keep extruding even here in cycles, and this is gonna work. Maybe rotate it this way, extrude this way, extrude this way, and then if we want to have like a nice background, maybe we can import a vesicle. So now we would be inside that vesicle. We can go ahead and hit 0 here. Maybe we want to rotate it in C the other way, that way, and then we can increase here the density to, let's go all the way to 10,000. So we go here to 10,000, then we're gonna have a nice background of the lipids there. And if we let the noise clear out a little bit, we can have a nice image of DNA within. This could be represented as a nucleus, or it could be just inside the cell, in the cytoplasm. And then this model that I made of DNA as a cartoon, as you can see, the geometry is pretty amenable to work with subdivision, so you could apply subdivision to this if you want to really zoom in. That way you don't get you get actual circles instead of this approximation. But yeah, that's the way you would get DNA into the scene, and also the way you would implement it into the workflow of generating these scientific visualizations. Next time we're just gonna look at the last part of it, that is gonna be viruses.